Biology has a set of rules. They are not written in a book, but they are fundamental. They are the principles we learn in school. DNA makes RNA. RNA makes protein. This is the central dogma. It is the flow of information in life. Genes made of DNA are the blueprints. They hold the instructions for everything. To build something, you make a copy of the blueprint. That is RNA. This copy goes to the factory, the ribosome. The factory reads the instructions and builds a machine. That machine is a protein. Proteins do almost everything in a living thing. They are the bricks and the motors. They are the messengers and the guards. They digest your food, carry oxygen in your blood, and contract your muscles. A protein's power comes from its shape. A protein is a long chain of smaller molecules called amino acids. This chain folds up into a very specific, complicated, three-dimensional structure. A key has a specific shape to fit a lock. A wrench has a specific shape to turn a bolt. A protein is the same. Its shape determines its job. If it loses its shape, it stops working. It becomes a useless piece of junk. The instructions in the DNA code for the sequence of amino acids. And that sequence, according to the rules, dictates the one correct functional shape the protein will fold into. Another rule of biology concerns infection. When we get sick from an infection, something has invaded our bodies. This invader is a living organism, or something very close to it. It could be bacteria, a virus, a fungus, or a parasite. What do all these things have in common? They all have genetic material. They have DNA or RNA. This is their own set of blueprints. They use these blueprints to make more of themselves inside of us. That is how they reproduce. That is how an infection spreads. An infectious agent, by definition, must have genes. It needs instructions to replicate. This was considered a law of nature. Then we found prions. And prions follow none of these rules. They tear up the biology textbook. They are a glitch in the system, a form of life, or something like it, that operates on a completely different logic. They are the exception to almost every rule we thought was universal, and they are terrifying. The story starts not in a modern lab, but with sheep. For centuries, shepherds had known of a strange disease called scrapie. A sheep with scrapie would become irritable. It would compulsively scrape its wool off against fences and posts, leaving raw patches of skin. Then it would start to stumble. It would lose coordination, tremble, and eventually, it would die. It was clearly infectious. If one sheep in a flock got it, others would follow. But no one could find the germ. The scientists looked for a bacterium. Nothing. They looked for a virus. Nothing. They took brain tissue from a sick sheep and treated it with things that should destroy any living organism. They used intense radiation, they used formaldehyde, they used heat that would cook any virus or bacteria into oblivion. But when they injected this treated tissue into a healthy sheep, that sheep still got scrapie. The infectious agent was impossibly tough. It seemed to have no moving parts, no life to extinguish. It was a ghost. Half a world away, in the highlands of Papua New Guinea, a similar mystery was unfolding among the four people. A disease they called kuru, which means to shiver or to tremble, was devastating their community, primarily affecting women and children. It started with an unsteady gait. Then came the tremors. Laughter would turn into uncontrollable, pathological bursts. Within a year, the victim could no longer walk, talk, or eat. It was always fatal. An American doctor named Carleton Gajdusek went to study kuru in the 1950s. He saw the similarities to scrapie. He also learned about the four's funeral rituals. They would cook and eat their deceased relatives as a sign of respect. Gajdusek suspected the disease was being transmitted this way. He managed to transmit kuru to chimpanzees by injecting them with brain tissue from a human victim. This proved it was infectious, but like scrapie, the agent was a mystery. He called it a slow virus, a virus that took years or even decades to show symptoms. It was the best guess anyone had. For a long time, this was the accepted explanation. The real breakthrough came from a young doctor named Stanley Prisner. In the 1970s, he had a patient with a rare brain disease called Creutzfeldt-Jakob disease, or CJD. It was shockingly similar to kuru. It caused dementia, loss of coordination, and death. Prisner became obsessed. He started to work with scrapie because it was easier to study in lab animals like hamsters. He spent years trying to purify the infectious agent from the brains of sick hamsters. He subjected his samples to every test imaginable. He used enzymes that chop up DNA and RNA. The agent survived. He used radiation and chemicals that mangled genetic material. The agent was unaffected. The only things that seemed to damage the agent were treatments that destroy protein. This led him to a conclusion that was at the time complete heresy. He proposed that the infectious agent was not a virus, not a bacterium, not anything with genes. He said it was just a protein, a single rogue protein. The scientific community was outraged. It was absurd. A protein is just a molecule. It cannot replicate, it cannot cause an infection. It has no instructions, no blueprint. To suggest otherwise was to challenge the very foundation of molecular biology. Critics called him a fraud. His grant applications were rejected, but he persisted. In 1982, he published his findings and coined a name for this new type of agent, prion. It was a mashup of proteinaceous infectious particle. So how does it work? How can a protein, a simple machine with no blueprint, make more of itself? This is where prions break the first and most important rule of biology. They do not replicate by building new copies from scratch. They convert existing copies. Your body has a normal prion protein. It is called PARPC. The SEC stands for cellular, its normal form. It is found on the surface of many cells, especially neurons in the brain. We are still not entirely sure what its normal job is, but it seems to be involved in protecting the brain and helping with cell signaling. PARPC is a properly folded, functional, and harmless protein. Everyone has it. The disease-causing prion, the rogue one, is called PARPC. The ECLES stands for scrapie, the sheep disease where it was first seriously studied. Here is the critical point. PARPC has the exact same sequence of amino acids as the normal PARPC. The chemical formula is identical. The only difference between the good guy and the bad guy is the shape. The good PARPC is folded into a shape with a lot of curly helices. The bad PARPC is misfolded into a much flatter, more angular shape with structures called beta sheets. This shape is incredibly stable. It is tough, like a little crystal, and it is a killer. The prions trick is a kind of molecular brainwashing. When a rogue PARPC protein bumps into a normal PARPC protein, it forces the normal protein to change its shape. The PARPC refolds into the toxic PARPSC form. It is a chain reaction. One bad prion creates another. Now, there are two. Those two find two normal proteins and convert them. Now, there are 4. Then 8, 16, and so on. It is a slow, relentless, exponential cascade of misfolding. The prion is not creating anything new. It is corrupting what is already there. It is like a zombie. It does not reproduce by giving birth. It reproduces by biting a normal person and turning them into another zombie. The information for this replication is not stored in a gene. It is stored in the shape of the protein itself. This is a form of inheritance that is purely physical, not genetic. That idea alone was revolutionary. This breaks a second fundamental rule that a protein's amino acid sequence determines one single, stable, functional shape. We thought the blueprint was everything. You follow the instructions, you get the product. But prions showed that the same sequence, the same list of ingredients, can produce 2 very different products. One is a useful cellular tool. The other is a stable, toxic, self-propagating monster. The information is not just in the sequence, but also in the fold. This means there is a layer of biological information that exists outside of DNA. The damage comes from the properties of this misfolded shape. Normal PARPC is soluble in water and can be easily broken down and recycled by the cell. It is disposable, but the misfolded PARPSC is not. It is insoluble and incredibly resistant to being broken down. The cell's garbage disposal system cannot handle it, so these rogue proteins start to stick together. They form long, tough fibers and clumps called amyloid plaques. These clumps build up in the brain. They kill neurons. We are not exactly sure how. Maybe they are directly toxic. Maybe they just clog up the cell's machinery. Or maybe the problem is the loss of the normal PARPC protein, which is constantly being converted. Whatever the exact mechanism, the result is devastation. The brain tissue starts to die. Under a microscope, you can see tiny holes appearing where neurons used to be. The brain literally becomes full of holes like a sponge. This is why prion diseases are formally known as transmissible spongiform encephalopathies. Spongiform means sponge-like. This leads to the third broken rule, the way these diseases originate. Most diseases are either genetic, meaning you inherit a faulty gene from your parents, or they are infectious, meaning you catch a germ from the environment. Prion diseases are both, and they can also be neither. They have 3 different ways of starting, which is unique and bizarre. First, they can be infectious. This is the kuru and scrapie model. You get the disease by being exposed to prion-infected tissue from another person or animal. The most famous modern example is mad cow disease or bovine spongiform encephalopathy BSE. In the 1980s in the UK, cattle were being fed supplements that contained ground up meat and bone meal from other cattle, including those sick with BSE. This transmitted the prions throughout the cattle population. Then people ate meat from these infected cows. In some rare cases, the cow prions were able to cross the species barrier. They managed to convert human prion proteins. This caused a new human prion disease called variant Creutzfeldt-Jakob disease, VCJD. This is classic infection. An external prion gets in and starts the chain reaction. Second, they can be genetic or familial. About 15% of human CJD cases are inherited. People with familial CJD have a mutation in the gene that codes for the prion protein. This mutation does not directly create the misfolded protein. Instead, it creates a version of the normal PARPC protein that is unstable. It is wobbly. It is much more likely to spontaneously misfold into the bad PARPSC shape on its own without any need for an external prion to trigger it. If it does misfold even just once in one cell, that single event is enough to start the unstoppable cascade. You inherit the vulnerability, the loaded gun. Sooner or later, it is likely to go off. Third. And most terrifyingly, they can be sporadic. This is the most common form of CJD, accounting for about 85% of cases. It just happens, out of the blue, for no reason we can identify. The current theory is that for a person with sporadic CJD, a single normal PARPC protein in a single neuron somewhere in their brain just misfolds by pure chance. It is a freak accident of molecular physics, a 1 in a million error. But in a system with billions of these proteins, 1 in a million chances happen. And with prions, one mistake is all it takes. Once that first domino falls, the chain reaction is inevitable. The disease appears to come from nowhere, so you have one disease that can be caught like the flu, inherited like cystic fibrosis, or just appear randomly like being struck by lightning. No other disease works this way. It breaks all the neat categories we use to classify illness. This brings us to another broken rule, the species barrier. Viruses are often very specific about who they infect. The human flu virus infects humans. The feline leukemia virus infects cats. Sometimes a virus can jump species, like the bird flu or swine flu, but it is often difficult. It has to adapt. Prions have a species barrier too, but it works differently. The barrier is based on the similarity of the prion protein between two species. The amino acid sequence for PARP is slightly different in a sheep, a cow, a mink, and a human. A cow prion will have a very hard time converting a human prion protein because the shapes do not quite match up. It is like trying to use a key from a Ford and a Toyota. It might not work at all. But if the sequences are similar enough, it might just barely work. The cow prion might be able to grab the human protein and force it to misfold. It is inefficient, but not impossible. This is what happened with BSE and VCJD. The cow and human PARP proteins were just similar enough for the jump to occur. The more similar the proteins are between two species, the weaker the barrier is. This is why transmitting prion diseases between primates is relatively easy, but transmitting from a sheep to a mouse is much harder. The barrier is not an absolute wall, it is a hurdle of probability. The existence of prions has forced us to reconsider what we mean by life and infection. A prion is not alive, it has no cells, it does not metabolize, it does not breathe or eat. It has no DNA. It is, in essence, a chemical crystal that can grow by converting other chemicals to its own form. It is a kind of gray area between chemistry and biology. It is an information parasite, but the information is structural, not genetic. And perhaps the most profound implication of prion biology is that this mechanism might not be as rare as we think. The core idea is a protein that misfolds and then causes other identical proteins to misfold in the same way, leading to clumps that damage cells. This pattern of protein misfolding and aggregation is not unique to CJD, scrapie, and kuru. It is the hallmark of a number of other much more common neurodegenerative diseases. Think of Alzheimer's disease. The brains of Alzheimer's patients are full of plaques. These plaques are made of a protein called amyloid beta. It seems that in Alzheimer's, amyloid beta misfolds and starts to clump together, much like the prion protein. There is also another protein inside neurons called tau, which forms tangles. Tau also misfolds and aggregates. There is growing evidence that this process is self-propagating. A small clump of misfolded amyloid beta can act as a seed, causing other amyloid beta proteins to misfold and join the clump. The problem seems to spread from one part of the brain to another in a predictable pattern over many years. It looks a lot like a prion disease in slow motion. The same is true for Parkinson's disease. In Parkinson's, the protein at fault is called alpha-synuclein. It misfolds and forms aggregates called Lewy bodies, which kill the dopamine-producing neurons in the brain, leading to the characteristic tremors and movement problems. Like the prion protein, misfolded alpha-synuclein seems to be able to spread from cell to cell, seeding new clumps as it goes. Amyotrophic lateral sclerosis, or ALS, is another example. Here, proteins like SOD1 and TDP43 misfold and aggregate in motor neurons, leading to their death and causing progressive paralysis. The mechanism appears to be the same. A misfolded protein corrupts its neighbors. Now, this does not mean that you can catch Alzheimer's or Parkinson's from another person. These diseases are not infectious in the same way as CJD. The protein aggregates involved seem to be much less robust than the nearly indestructible PARP-PSC. They do not seem to be able to survive, for example, the digestive tract or routine sterilization. But the mechanism of spread within an individual patient's brain is strikingly similar to a prion. Scientists now talk about prion-like behavior or prionoids to describe these other proteins. Prions are the most extreme and transmissible example of a much broader phenomenon in biology, the toxic propagation of protein misfolding. This has completely changed how we approach these diseases. Researchers are now looking for ways to stop this seeding process. Can we develop drugs that stabilize the normal protein so it cannot be corrupted? Can we create antibodies that seek out and clear away the misfolded clumps before they can spread? Understanding prions gave us a new framework, a new model for thinking about the root cause of some of humanity's most devastating and untreatable brain diseases. The rules of biology are powerful because they are so consistent. DNA makes RNA makes protein. Shape equals function. Infection requires genes. These principles govern trillions of organisms every second of every day. And yet, in a dark corner of the biological world, prions exist. They are a testament to the fact that life, or something that mimics it, can find a way to propagate using a completely different system of logic. They show that information, the very essence of life, does not have to be written in a genetic code. It can be encoded in a physical shape, a twist of a molecule. A prion is a whisper that can turn a healthy protein into a killer. It is a typo in the folding, not the script, and this typo can be passed on, a message of destruction that carries no letters, only a deadly shape. They are simple, elegant, and completely alien to the way the rest of life works. They are biology's broken rule.